So where did it all start? Aloysius Alzheimer was the first scientist to submit a scientific paper regarding Alzheimer's disease back in 1901. So this disease has been discovered for about 118 years. But essentially, there have been no significant progress in treating Alzheimer's disease, and it's becoming more prevalent worldwide. And so, to combat this increasing rate of Alzheimer's disease, in the Aging and Vascular Physiology Lab, we believe that understanding the vascular system is key to creating potential treatments for Alzheimer's disease. And so, I'm specifically looking at something called the blood brain barrier, which is part of your neurovasculature or system of blood vessels in your brain. Now, the blood-brain barrier divides the blood from the extracellular environment in which the neurons reside in, and it's normally a very restrictive barrier in that it allows very few substances to pass through the barrier. And this makes sense because we don't necessarily want any neurotoxic compounds to enter our brain tissue. Now, when you age, your blood-brain barrier may potentially start to break down and become leaky. And so, if you look at figure one, this explains why this could potentially be a problem. So, neurotoxic compounds, red blood cells, that could actually make its way into the brain tissue, leading to inflammation. And inflammation can actually induce the formation of amyloid beta plaques, which is normally what we see in Alzheimer's disease patients. So, to combat this increase in permeability, we believe that the Robo4 protein receptor can inhibit an increase in blood brain barrier permeability by inhibiting vascular endothelial growth factor, therefore inhibiting angiogenesis or the formation of new blood vessels. So, when you actually form new blood vessels, you actually would have to increase the permeability of those existing blood vessels, which is not necessarily what we want in this case. And so my experiment involved using young and old wild-type mice. So we use C57BL6 mice and Robo4 knockout combined with a mutation called APP, which stands for amyloid precursor protein. And this mutation leads to an overproduction of amyloid beta. Now, normally mice do not get amyloid beta plaques in their brain. So we actually have to genetically introduce that through APP mutation. And so we hypothesized that old mice and Robo4 knockout mice will exhibit greater cognitive decline. And to measure cognitive function, we used nest building tests and Moore's water maze. So in a nest building test, we basically placed each mice into separate cages with a paper nestlet and we left them overnight. And so normally on mice, they would create a nest that is circular, it contains a crater, and is higher than the mice's body. And so we rated each nest through a scale of 0 to 5, 0 that no nest was formed, and 5 being that a perfect nest was formed, making its way into the Moore's water maze. So this test was conducted for 5 days, and the main objective was to allow the mice to determine the location of the platform. And so on day 3 and 5, we removed the platform to measure how long the mice stayed in the quadrant of where the original platform was located in. And this will allow us to determine whether the mice learned where the platform was. Going to the results, figure 2, it shows for part A that at day 3 of the experiment, there was no significant difference between young and old mice in terms of how long they were in the target zone. However, on the last day of the experiment, part B, the young mice significantly had a greater amount of time in the target zone than the old mice. And this demonstrates that the young mice were able to remember where the platform was better than the old mice. Figure 3, so part A represents the nesting scores between the old and young mice. So the young mice had a significantly higher nesting score than the old mice. And part B represents the nesting scores between Robo4 wild type, knockout. So a knockout is that this Robo4 protein receptor is not present. We also did Robo4 wild type with APP mutation and without APP mutation. We found that there was no significant difference with Robo4 deletion or amyloid beta overproduction. So therefore, we can conclude that old mice had impaired cognitive function when compared to the young mice. However, there was no significant difference in cognitive function with Robo4 knockout or APP mutation on nest building tests. And so we need to conduct more cognitive tests on Robo4 mice crossed with the overproduction of amyloid beta. And we will also measure blood-brain barrier permeability as well as to examine the formation of amyloid beta and neuroinflammation in Robo4 mice. So although this set of studies did not find a role for Robo4 in cognitive impairment, we need to perform further studies before making definite conclusions. So our continued studies in this area will help us better understand the role of the blood-brain barrier permeability in Alzheimer's disease. And that's all I have for you. Thank you.